Welcome to the webcast on improved analysis of 1,4-dioxane in drinking water. The authors are Melissa Lever and Michael Ebbetson of Horizon Technology, co-authored with Phil Bassignani from Alpha Analytical in Mansfield, Massachusetts. The physical and chemical properties of 1,4-dioxane are shown in the following table. You can see that it's water miscible. It also is fairly volatile, two things that make it a little bit more difficult to get it out of water and retain it for analysis. "1,4-dioxane has been shown to have health effects, which is why it's a compound of such concern. The US Agency for Toxic Substances and Disease Registry thinks that dioxane at high levels may cause liver and kidney damage. The Department of Health and Human Services has stated that 1,4-dioxane is reasonably anticipated to be a human carcinogen based on evidence of carcinogenicity in experimental animals. EPA has also classified 1,4-dioxane as likely to be carcinogenic to humans by all routes of exposure. Outside the U.S., the World Health Organization classifies 1,4-dioxane as a Group 2B compound, which means that it's possibly carcinogenic to humans in their view. The Agency for Toxic Substances and Disease Registry has a substance priority list. On this list, they rank compounds by their toxicity using a point system. 1,4-dioxane is on this list at position 214 and has accumulated 617 points of toxicity measurement. The most toxic compound on the list is arsenic with 1,674 points. Now, this list is also used as a basis for measurements at the EPA National Priorities List sites. So, for example, it's been found at more than 34 sites on that list. However, not all samples were measured for that compound, so it may in fact be present at other national priority sites as well. So we've seen that there are some health effects expected from 1,4-dioxane, and we've talked a little bit about some occurrence where it's been found, but where does it originate? Well, sources of 1,4-dioxane include its widespread use as a stabilizer in certain chlorinated solvents, and it's often found in conjunction with those chlorinated solvents. It's a byproduct present in many goods, including cosmetics, and in fact the EU Scientific Committee on Consumer Safety has recommended a limit for 1,4-dioxane in finished cosmetic products, and they would like that to be less than 10 parts per million. So you can see from some of these sources that we all have the opportunity to be exposed to this compound in many different ways. Some of the ways that we can be exposed to 1,4-dioxane are more impactful. So things such as food supplements, food containing residues from packaging adhesives, or on food crops treated with pesticides that contain 1,4-dioxane may have a larger health effect. Certainly the air we breathe can be an issue, recreational waters that we may swim in, and drinking water, which provides a very large source of exposure of any contaminants. The US EPA has the responsibility to keep our drinking water safe, and as such has created a candidate contaminant list, which is continually developed to list compounds of emerging concern for further investigation. Every 5 years, 30 of those analytes must be evaluated further to see whether or not any sort of regulation is merited. So toxicity is part of that, but they also need to understand whether or not it occurs in drinking water sources. So the Unregulated Contaminant Monitoring Rule, UCMR, is used to assess the occurrence in large and small drinking water supplies of candidate analytes that arise from the candidate contaminant list. The third UCMR collected data on 1,4-dioxane from 2013 to 2015. "1,4-dioxane" in UCMR 3 was written up extensively in this paper included in Science of the Total Environment. And this paper had a number of really interesting charts in it showing how 1,4-dioxane was seen around the United States. The paper included this really nice map of the United States mainland showing 3 different concentration levels for 1,4-dioxane measured in UCMR 3. The open dots are actually below the minimum reporting level, which is 0.07 micrograms per liter. The gray dots are above the minimum reporting level but below the reference concentration, which is actually the health advisory level, which has been set to 0.35 micrograms per liter. And then the red dots are the highest level that was observed, anything above that reference concentration or health advisory level. So it really gives you a snapshot of everything occurring at one time. There were also some observations in other parts of the U.S., and in fact 50 of the 63 states, districts, territories, and tribal areas that conducted UCMR monitoring had some detections of 1,4-dioxane. We just mentioned that there have been some health advisories set for 1,4-dioxane, and this table summarizes the information of drinking water standards and health advisories and was published in March of 2018. So you can see that 1,4-dioxane is listed here with a number of different things. There's no regulatory limit mentioned, but there has been a health advisory recommendation of 0.35 micrograms per liter in order to avoid a 1 × 10⁻⁶ cancer risk. Some of the states don't want to wait for regulation on 1,4-dioxane because there's concern amongst their constituents. So you can see a list of states with guidelines listed for the measurement of 1,4-dioxane. Most of those are higher than the advisory limit set for drinking water. A number of EPA methods have been evaluated for use for different matrices and measurement of 1,4-dioxane, such as the volatile methods 524, 624, and then also the RECRA methods 8260, 8270. And so far it seems that US EPA Method 522 for drinking water has been shown to provide reliable results and the lowest detection limits that could be measured to date. So this talk is going to describe the implementation of Method 522 in a commercial laboratory incorporating automation to improve method performance. Here you can see Method 522 set up in a commercial laboratory. This is the extraction step. Cartridges are used for extraction of the 1,4-dioxane from drinking water, and they need controlled flow for the best performance. So therefore automation is included, and this also provides less technician interaction, less potential evaporation of the volatile analyte, less chance of introducing contamination. So the method is automatically, reproducibly implemented, which is especially good for a multitasking workforce. The extraction method is shown in this table. You can see that there are 12 steps, but since it's automatically implemented through an automated system, the technician doesn't have to worry about remembering what the order of steps is or exactly how much solvent to pour onto the cartridge. The important steps to look at are 10, 11, and 12, where you can see that a total of 9 milliliters of eluent is collected with the analyte in it. And that's a very small amount of eluent, and no further evaporation is done in order to best retain the 1,4-dioxane. Here you see the GCMS conditions used for this method. A Phenomenex Zebron ZB5 column is used for the GC. The mass spec is used in single ion monitoring mode. So you can see tetrahydrofuran D8 is the internal standard, 1,4-dioxane D8 is the surrogate, and 1,4-dioxane is measured at 58 and 88 atomic mass units. Method detection limits were calculated based on 8 replicates of a 0.1 micrograms per liter standard. You can see the calculation here. A method detection limit of 0.01 is actually pretty respectable when the minimum reporting level is expected to be 0.07 micrograms per liter. The method detection limit was confirmed by calculating the upper and lower limits for the prediction interval of the results. And you can see in the table listed here that in fact both the upper and lower limits pass the criterion listed in the method. The reporting level used by a laboratory can be determined this way, or the lowest point on the calibration curve, if greater than this value, can be used. In addition, spikes were done at low levels to ensure that the method was under control. Five mid-range laboratory fortified blanks were used for the initial demonstration of accuracy and precision. The criterion for accuracy is that the mean recovery is plus or minus 20% of the true value. The true value is 50 micrograms per liter, and the recovery in this case was 91.1%, well within that acceptable window. The precision must be less than 20%, and 11.3 for the RSD is better than the requirement. So both requirements were met for the initial demonstration of accuracy and precision. In this slide, we're looking at our lab fortified blank, both at the low, medium, and high levels, and looking at the percent recovery. The percent recoveries were well within the 50 to 150% recovery of the true value, and the surrogates were within the 70 to 130% recovery in all cases. So the analysis was under control when those were measured. In this particular quality control test, we're looking at the continuing calibration check results, both low and high, and you can see that the measured value is within the requirement for recovery of the true value. It must be within plus or minus 50% of the true value, and it's 103% for the low continuing calibration check, and 94.5% for the high continuing calibration check standard. In the method, a 2-gram coconut charcoal cartridge is suggested. In addition, a 3-gram coconut charcoal cartridge was evaluated. The 3-gram cartridge allowed faster sample processing of the 500 ml sample and allowed better surrogate recoveries. Fewer reruns were also observed, less than 1% over the course of several months. So the 3-gram cartridge is definitely an advantage in improving performance of this method. The improved surrogate recovery of the 3-gram cartridge is shown in this chart. You can see across the x-axis 19 different samples and the surrogates measured, and percent recoveries on the y-axis, and the recoveries are all between 80 and 100%. So the reproducibility has been improved, as well as the increase in the percent recovery. We all know that a performance evaluation sample provided by a third party and run as a blind sample is the best way to evaluate whether or not the method is truly working in a routine situation. In this case, we can see that the measured value of our PT sample was 7.82 and the true value 8.0. So actually the percent recovery is 97.8, which is excellent and well within the acceptable range of that particular study. To conclude, the method developed meets the quality control requirements specified in US EPA Method 522. In fact, the method was improved by going to a 3-gram cartridge rather than a 2-gram cartridge so that additional sorbent was available to allow the sample to be put through more quickly and to better retain the analyte and surrogate. Automation of the system provides less technician interaction required, so there's less potential evaporation of a volatile analyte or less chance of introducing contamination. For laboratories that run many methods or run this method infrequently, automation enhances reproducibility. It really makes a difference to have automation for a cartridge. You need to put the sample through at a very controlled rate, so having it done through an automated process eliminates the variability between technicians. Use of a system that can switch between sorbent discs and cartridges increases system usage and efficiency in the laboratory workflow. This is particularly important for laboratories that run a variety of methods and projects. Now, this method may be extended in the future to other types of water, including those with small amounts of particulate. So overall, it was a very successful implementation of Method 522 and could be extended in the future. Thank you.